Bismillahirrahmanirrahim, and welcome back to this next video. And this is the last video of the first chapter of the bioinformatics. In this particular chapter, I generally focused on the structure of the DNA, structure of the RNA, and the structure of the protein. A brief history of the bioinformatics, how it started, and how the bioinformatics that is helping the understanding of the life sciences at the molecular level, and how it can help you in the different experimental procedures of the biology. So before this video, I generally talked on focusing on single genes, because when you look at the history of the bioinformatics, they usually started with proteins first, then they moved towards the sequencing of the DNA, and they were mostly focusing on a single protein or a single gene. Now, in the current era, we are actually working with the entire genomes. So this video is going to focus on how the work on the entire genome that started. So the first truly efficient technique to sequence DNA that was discovered in 1977 by Frederick Sanger, and using this technique in 1995, the first sequence of an entire genome of a bacteria known as the Hemophilus influenza that was determined with this DNA sequence of like 1830140 base pairs of DNA. So this was the first complete genome sequence that was available to the mankind. Now, between these years, like 1977 and 1995, the DNA sequencing technologies, they improved steadily, but these technologies that were mainly focusing on mining individual genes for information, and that was logical because they haven't mastered the technique yet. So they were focusing on single gene, and the single gene, they are made up of a few thousand base pairs, so you can easily work with them. So the focus of these particular technologies in the beginning, that was mostly on sequencing DNA fragments that were like a few thousand nucleotide in length, simply because they were interested in specific genes. The scientists, they were focusing on single gene, which was the subject of their research. So they'd start sequencing those particular specific genes that had worked starting on years before. Now, most of the bioinformatic tools that are available today, that were created during that period, and that includes the sequence alignment programs, all of the basic sequence alignment programs. Don't worry about the basic sequence alignment programs. What are they? I'll be focusing on them in detail. The phylogenetic and the classification methods, and the various display tools that were adapted to relatively small sequence objects. For example, if you're dealing with a protein sequence, no more than a few hundred amino acids. Now, when you talk about the genomics is actually the field where you focus on whole of the genome. And the genome simply means the entire genetic content of a living organisms. So in genomics, you are getting all of the gene at once. So the determination of the first complete genome sequence of the Hemophilus influenza, that actually terminated the gene by gene routine. So now the scientists, they were not focusing on single genes. They actually started the era of the genomics, where they were focusing on all of the genetic content of an organism, like the genetic mapping, the physical mapping, and sequencing of the entire genomes. Now, as a consequence, the DNA sequence, which are now available in different kind of the databases, they are like of million base pairs in length for the microorganisms, and up to several billion base pairs in length for animals and the human beings, because these animals and the human beings, they have got big genomes as compared to the microorganisms. Now, this revolution, they called for design of new bioinformatics tool and databases capable to store, query, analyze, and display these huge objects, because if you're dealing with like billion base pairs of the DNA, that means you're dealing with very huge objects. So they designed a new bioinformatics tool so that you can work with these huge objects in a user friendly manner. Now, in contrast to the early gene by gene approach, the DNA sequences are now often obtained without any prior knowledge of what is actually there. Because in essence, you can say that the gene these days, they are both sequenced and discovered at the same time. Now, this development, where you are like sequencing and discovering the gene at the same time, has prompted the emergence of an entirely new branch of bioinformatics, which actually focus to resolve the large DNA sequences into their components. Like when you talk about the larger DNA sequences, you can have the genes, you can have the transcription units, the protein coding region, the regulatory elements, etc. So, bioinformatics is actually focusing on resolution of these particular components. This is then followed by a longer phase of genome annotation. And this genome annotation, what it do is the biological function of these various elements, they are predicted and they are tried to find out. So this is the end of the chapter 1 of the bioinformatics. So if you like the videos, please subscribe to my channel, hit the like button and share it with your friends. And in chapter 2, I'll start working with the PubMed, how you can work in the PubMed, how you can find out your specific genes or your proteins. So I'll focus on that in my next video.